I'm not going to talk about how motility is controlled. This has to do with the key system that you've all learned about. And in general, in bacteria, some modified form of the key system regulates motility throughout the bacteria. Whether that motility, no matter what the mechanism of motility is. We're talking here about the mechanical aspects of it. What does an organism do physically to move from one place to another? And so, you all know about flagella. A flagella is basically a proton gradient-driven propeller. And so there's a little motor here, essentially an electric motor. It's driven by flow of protons from one side of the capacitor membrane to the other. That helical rotation spins this flagellum, and that flagellum then pushes the cell in the opposite direction. This is different than flagella in eukaryotes, which are quite complicated structurally. There's a bunch of tubulins sliding next to each other. And most flagella in eukaryotes pull rather than push. In bacteria, they push. Now, this is the simple system where there's a single polar flagellum. There are lots and lots of variants of this. Usually, it doesn't matter if you've got lots of flagella. When they spin, they tangle up and form a much larger helix, superhelix of a cluster of fibers rather than a single fiber. And it works that way whether they're all polar or whether they're surrounding the cell or anything else. Now, you'll notice that this is a rod-shaped organism, more or less. Most flagellated bacteria are either rod-shaped or a modification of that. It's uncommon for cocci to be motile by flagella. In fact, it's uncommon for them to be motile at all. But a rod-shaped organism, although it's more hydrodynamically favorable than a coccus, there's a lot of loss associated with motility of a rod-shaped organism. Because as this flagellum rotates, it doesn't work unless there's resistance against the environment, right? That resistance to rotation causes the cell body itself to counter-rotate. Imagine a submarine without ballast at the bottom. As the propeller spins, the whole submarine would spin too, right, in the opposite direction. And that would be loss of energy. Wouldn't it be great if you could recapture some of that energy? Well, you can do that by making the cell a helix too. And so as this thing spins and goes this way and pushes, this thing counter-rotates and it's a screw too. And the result is that rotational energy isn't lost, but is captured also in motive force. And so lots of organisms have done this. We call these spirilla when they're driven by flagella this way. This has lots of ramifications for how motility is controlled, and these organisms typically do a tumble and run motility, and these ones will typically have polar flagella at each end, and instead of tumbling, they will just turn off one set of flagella and turn on the other to back up. And there are other ways that you can control that sort of thing, but again, that's control, that's not the physical aspect of it. But not all bacteria move by flagellar rotation. Another mechanism is gliding. And gliding isn't really a mechanism, but it's a catch term for a variety of motility mechanisms that end up with the cells moving smoothly, or not so smoothly sometimes, over a surface without flagella. So it's kind of by an exclusion process. There are a variety of mechanisms for gliding motility. A pretty large fraction of them do it by the secretion of polysaccharide from pits in the cell. So imagine a rod-shaped organism with pits at each end, and polysaccharide is secreted into that pit through the periplasmic space. When a polysaccharide is pushed through a membrane, it's dehydrated. And so when that polysaccharide hits the environment on the far side in that pit, it will immediately become hydrated. And polysaccharides bind a bunch of water, right? Typically, a polysaccharide will bind between 20 and 100 times its volume in water. That's how a rocket engine works, right? You secrete a small amount of stuff, a small volume of stuff into a space, and that expands. But it's in a pit so that that expansion is directed. And the result is each one of these is a little rocket engine. And the cells move along. And they leave behind them a trail of hydrated polysaccharide, something that is scientifically called slime. This is how snails move, right? And they leave behind a little trail of stuff. This is how diatoms move. This is how most cyanobacteria and lots of other gliders move. This works even in a filament, because in a filament, each cell has pits on the leading and trailing edge directed in that appropriate direction and cause the entire filament to move by gliding. Some gliders are capable of moving over surfaces only, but most gliders that use this mechanism can even do this in an aqueous environment. And so they leave behind a string of slime through the environment. Another form of gliding, the bacteroids do this, and others as well, is basically a tractor motility. This is a lot like the tractor tread on a crane or a tank or something like that. And so an adhesin is pushed through the membrane at the leading edge of the cell, and it's attached to the cytoskeleton of the organism through the membranous structure and attached to the cytoskeleton, which of course is a helix around the surface, around the inside of the cell envelope. And that adhesin then is pushed with little motors down that cytoskeletal fiber to the tailing end. And so since this quickly touches the substrate and sticks there, as it's pushed along the helical cytoskeleton, the cell is driven forward. And then at the trailing edge, it's pulled away from the substrate, internalized, and somehow makes its way back to the beginning again, and this process repeats over and over and over again. Gliding motility in mycoplasma, there's a variety of mechanisms for this, but one of them is really simple. There are little adhesion legs that bind and pull backwards. But how that works is a little bit unclear. A variant of gliding, and sometimes this is called gliding, is twitching motility. Whether it's called gliding or twitching depends on what it looks like under a microscope. So classical twitching motility, Pseudomonas does this, is that you have pili extended from the leading edge of the cell, and they extend out. Pili are built by adding monomers to the fiber from the internal side, the proximal side of the fiber. And so it reaches out, there's an adhesin at the end of it, and when it touches the substrate, it sticks. And then the pili is retracted, pulled back in again. And the result is, if it sticks here, and then this is shortened, the cell pulls forward. If it uses one or two pili at a time, they really are twitching. The cells will yank forward, in kind of a random way in some general direction. Again, control whether the cells, continue to secrete pillins from this end or do it from this end is controlled by the key system. If, on the other hand, the organism produces lots of these pili all the time, then the combined effort of 20 or 30 of these acting not synchronously but haphazardly all the time is that the cells move pretty quickly and pretty smoothly. And very often this is called gliding motility, even though the mechanism is different than the normal gliding mechanisms. And so what you saw for the Synechococcus on Friday was a kind of twitching motility. One of the forms of motility that people usually forget about is gas vacuoles. And so this works in aquatic environments, and it's particularly common for phototrophs. So if you're a phototroph floating around in a lake or in the ocean, it's really important to you to be able to find a place where the light intensity is optimal, right? Maybe that's right up near the surface, maybe it's somewhere else, depending on your other requirements. And these organisms are quite often modeled by the production of gas vacuoles. Got a couple of scuba divers in here. You know how buoyancy control works. You pump is usually a CO2 in these gas vacuoles. These gas vacuoles are usually spindle-shaped protein structures, and the organisms pump CO2 in or let it out. This CO2 comes from metabolic activity, usually it's CO2. And they use it to control buoyancy. That is, if you want to move up in the water column, you pump more gas in, and if you want to sink, then you pump gas out. And the gas that pumps out gets converted into bicarbonate, which is secreted. Yeah.
You said the backwards is
Usually the outside, the shell is protein. Yeah, so it's flexible. There are a few instances where there are membranes, lipid layers for that, but it's not very common. Usually they're protein. This has to be very, very tightly regulated. Again, those of you who are divers know this. When you're neutrally buoyant, that has to be an active process, because any little perturbation, maybe a current that lifts you a couple of feet into the water column, all of a sudden now you're not, if you move up in the water column, now you're more buoyant than the surrounding water, and you'll start to rocket to the surface. And the more closer you get, the more you rocket up, because you expand as that pressure is released. These gas vacuoles will physically expand, and the cell becomes less and less buoyant the higher up in the water column it goes. Likewise, if the cell is pushed down a couple of feet in the water column, that increased pressure will squeeze these, decrease their volume, and the cells are now more dense than the environment, and they'll sink. And the more they sink, the less buoyant they are, and the process is unstable. And so the organisms have to constantly monitor their environment and constantly adjust their buoyancy to stay where they need to stay. This is microcystis, by the way. That's the green slime stuff. This organism likes to float right on the surface. And so that's why there's so many gas vacuoles in this thing. We talked about spirochete motility. There's a modification of this that's found in Spiroplasma. Spiroplasma is not a spirochete. Spiroplasma, they're members of the firmicutes. They're related to mycoplasma, common plant pathogens and plant symbionts. They're helical, but the axial fiber is not made of flagellum. It's actually a series of fibers that form a band, and it's a kind of linear motor against each other. So they're able to twist and manipulate the length and shape of that band at will. They don't actually rotate. What they do is they're helical shaped, and they can flip at any place along their length. They can flip from a right-handed turn to a left-handed turn. Think of this like an old-fashioned telephone cord. It's a helical thing, and usually partway down the cord there's a place where it reverses direction and goes the other way again. Now imagine you could create one of those anywhere in this length and then move it. And so they would create a reverse polarity here at the end, and then push that reverse polarity all the way down from one end of the cell to the other. That creates a grip, right? It can't turn freely in the environment anymore. And so that creates a grip on the environment. And when that pushes down the cell moves forward. And typically for a long spiroplasma, you would create 2 or 3 of these down the length, and they're all moving down the cell as they go. And that creates a motive force to push them. In this case, usually between the cells of a plant, or within the cytoplasm of the plant, depending on its exact environment. These are 7 pairs of fibers that create this really interesting motor system. Oops. Yeah, so this picture is not that great. You can see they're doing a flip here at this end, where it flips polarity, and then it pushes down. It's not real clear in this picture, but it pushes down the cell from one end to the other. Yeah. It's just the end of this entire one. all of this is to get around the fact that if you just have a plain helix and try to turn it, you have to figure out some way to convert that into motive force. It's not